All right, so now we're going to start part C of the first chapter of the Pierce text, so Introduction to Genetics. And what we're going to talk about now is still talking about a historical perspective, but we're going to look more specifically at Gregor Mendel. So, Gregor Mendel was a, uh, a monk, and he discovered the basic principles of heredity. So, and this happened in about 1850, and he actually um, started to put the scientific method into process. So, researchers had before him, like Joseph Krutler in about 1800, and even folks further or earlier than that, used to just make observations, but they never used to repeat studies. So, what Mendel did, and we're going to study his work a little bit more in depth when we get to chapter 3, but he actually created genetic models, and he selected his own parents. In or he selected his own parents, and he used the pea plant as his genetic model. So he was able to identify pure pea plant parents. That was hard to say. So that he knew exactly what those genotypes were going to be. And so his discovery and his work there at the monastery actually really led to a variety of new discoveries in genetic technology. So everything from cell theory to the structure of how this genetic information was stored and passed down from generation to generation. And his work really um, wasn't very popular until for about another 50 years. So, as we think about the changes that genetics have actually affected, and how DNA structure has been affected through not only selection but natural selection as well, we've been able to create food products that yield higher amounts of protein and nutrients, are disease resistant, pest resistant, and have enhanced nutritional value. So that last point was really culminated in 1970 by Dr. Borlaug, who received the Nobel Prize for the hybrid variety of rice that was developed. So he took, some or a rice strain um, that had a lot of survivability characteristics, crossed that with um, another rice strain that was more productive, and they were able to create this new hybrid model. So biotechnology has advanced much more rapidly since that time, and we've been able to develop different biotechnological procedures in a variety of new ways. So one of the ways that biotechnology has actually helped us is by the ability to actually detect defective genes, like we talked about in the osteoporosis or the arthritis example. So, us being able to identify where the gene actually lays in the chromosome allows us to assess a location for treatment on that chromosome as well. So, these biotechnology procedures really open the door to pre implantation genomic screening. So, the ability for a mother to go in before a child has even been born and have a DNA test ran on it to determine if everything is okay or if there's some sort of genetic abnormality. Other things that we've been able to utilize in biotechnology is staining and development um, of embryos, and how. So what you're looking at right here is actually a fly egg, and or a fruit fly embryo, and the way that these embryos actually develop is that depending on the location of the larva, that certain area will result in a certain body part. So the Peter Lawrence in 1992 was able to go in and actually stain specific cells within the larva, and they were able to track what body parts of the fly that those different areas actually turned into. So in this example, we've got two colors, the gray color and the brown color, and it kind of looks like blue on this. And those are going to determine respective body parts in the adult fly. So another example is cloning. So cloning first became popular with the sheep in 1997. That was done at the Roslin Institute in, I'm going to get this wrong, Switzerland, I think. Yeah. No, maybe Sweden. Anyway, no, it's Scotland. I'm sorry. So Dolly the sheep was cloned in 1997, and since then we've been able to clone a variety of different animals. So one of them is actually Cece. So Cece is this kitten shown in the top two photos here. And Cece was a cloned offspring, and she was born in 2001. So the ability to take a somatic cell and insert genetic material into that cell, and then allow that embryo to go through the development stages is a pretty awesome process. So I've been involved with a little bit of cloning that I'll talk a little bit later as we roll through this class about. And then gene therapy is really where a lot of our technology is at today. So the ability to treat conditions by using therapeutic measures at the DNA and the gene level. So the way that gene therapy works is that we have a person that has some sort of phenotype that isn't desirable, so some sort of disease. So their cells have the not ideal version of the gene. So what we can do is take those cells and create a virus that contains a functional gene and allow that virus to be uptaken into those affected cells. And once that virus is uptaken, it changes the phenotype of that cell, and the individual is then infused with these infected cells. They'll be taken into the body and start to reproduce and produce normal phenotypes. And that's using two different mechanisms. So the individual's own cells and then a virus as well. So gene therapy was utilized in the first patient in 1990. So this is technology that actually really isn't very new, even though when we look at the historical perspective, it is fairly new. So some of the diseases that can be treated with gene therapy are ADA-SID. So this is like the autoimmune disease where an individual doesn't have any immune system at all. You guys may have seen the really poor Bubble Boy movies, but these were actually based off of true stories. So other diseases are like hemophilia, muscle dystrophy, congenital blindness, lysosomal storage disease, certain cancers like leukemia, and then neurodegenerative diseases. So Parkinson's as well as Huntington's disease. And now we're starting to see some gene therapy happening with Alzheimer's disease. So in the 20th century, we really saw a huge increase in biotechnological advances as we moved from the 1800s into the 1900s. So Mendel published his work in 1866 that was using peas. By 1902, Walter Sutton really came up with this concept of genes being located on chromosomes. So quite a few years in between there. And then by 1930, the concept of population genetics, or studying how the prevalence of genes in certain populations, was born. In 1953, so a short 23 years later, Watson and Crick did a big lit review and was able to pull together that DNA was actually a 3D structure. So they didn't do any of the lab work, just want to put that out there, but they did do an extensive literature review to make that determination, and that was published in 1953. And then things start to get busy very quickly. In 66, we knew that DNA was a chemical substance. And then in 73, we actually made the first recombinant DNA. And Shortly following that, a mere 4 years later, Gilbert and Dr. Sanger first sequenced DNA. So they actually were able to sequence out the amino acids that were creating certain genes. One of the most phenomenal pieces of technology that we still use today, polymerase chain reaction, was developed in 1983, and this was used to clone specific genes in a high frequency. And then 1990, we know that the first human was treated with gene therapy. And by 1995, we had fully developed genome sequences for certain species. So the future of genetics is we're really just looking at the tip of the iceberg. So new discoveries are being found at a really an increased speed and an increased rate. And we now know that a lot of new information when it comes to genetic research actually requires a computer. So there's this whole discipline called bioinformatics that blends computer networking systems with genetic information. So, as we begin to not only finalize the human genome sequence, but finalize that sequence in multiple individuals from multiple backgrounds, we're going to be able to genetically treat diseases based on genomic sequences. So it's a very, very exciting time with genetic information and research at this point. All right, that concludes part C. You guys have one more part to get through, and then that completes all the material for chapter 1.